last time we were talking about genome engineering applications, and we shifted from the idea of potentially knocking out many different instances of one particular type of gene, like those that encode aldehyde reductases. And we then discussed genome recoding, looking at another aspect of the genome that could be redundant because of the degeneracy of the genetic code, where we might want to enable a new chemistry by removing, in this case, a codon from the genome. And we discussed some efforts in the field to do that, first using multiplex automated genome engineering techniques, and then more recently, the trend towards using DNA synthesis to design and construct entire genomes from scratch, as the cost of DNA synthesis has decreased substantially over time. So we left off with this example where there were less than 61 sense codons. And in general, this idea of trying to do this kind of genome recoding is that of freeing up codons. They may be reassigned later to non-standard amino acids, but for now, we consider these sort of less than your 61 sense codons. What I want to shift gears into talking about, which is still very related, is another kind of genome engineering that's ongoing, that's fairly impactful. As kind of shown here at the bottom, there was a startup called SynthWorks that was really pioneering this space, and they were recently acquired by Sanofi, a biotechnology company, for $2.4 billion. So highlighting kind of the importance or potential of some of these technologies. So you can have more than 61 sense codons. And their approach to doing this was to not rely on the standard genetic alphabet, which consists of your A, T, G, C, but rather to take work that had been explored for a number of years, it's fair to say decades, but in different contexts, usually in vitro, screening different kinds of artificial base pairs, looking to see if they can form Watson-Crick base pairing or other kinds of bonding, including hydrophobic interactions. And one of those base pairs, which was for simplicity nicknamed X and Y, was then integrated into the actual DNA that could replicate within a living organism. Of course, it was tested out in E. coli. And so you had an organism that had a genome in which, now instead of your 4 nucleotides, which because of triplet codons give you 4 cubed, leading to 64 different codons, you would now have 6 cubed number of codons that you could generate. And so I think this is just showing the before here, and how you could go from 20 amino acids to then, now when you have this XY pair, you have 6 nucleotides, you can get up to 216 codons, which can encode 172 amino acids. And I don't want to necessarily spend too much time on, there's a lot of aspects to this. I mean, of course, you've got to be able to have your artificial base pairs, not just at the DNA level, but also at the RNA level. You've got to have a way for them to get into the cell. You've got to have a polymerase that can, form RNA and accept the RNA. You've got to have the right corresponding tRNA and all of that. But suffice to say that if your goal is to introduce multiple amino acids that don't exist naturally or that aren't one of your standard 20 building blocks, then this kind of intervention at least at the DNA level, does not require that you modify the entire genome. You just have to insert your X and Y and do that. Now, this is an oversimplification, but in, say, one gene, without necessarily worrying about doing some kind of synonymous substitution throughout the genome. Now, it is the case that because you've got to enable their import, the cell can't make those bases on their own, and you've got to also engineer the mRNA, the tRNA, and then eventually the aminoacyl tRNA synthetases. It's not trivial to do this either. But you can imagine this kind of approach scaling in some ways better than recoding if your primary or only goal is to add more amino acids to the genetic code.